Hello champions and welcome to my playlist of Cambridge O-Level Biology 5090 that we are studying from Hodders biology book, and today we are going to start the first chapter, which is about cells. So the blue box of this chapter says to focus on differences between animal, plant and bacterial cell. These are extremely important topics for your Cambridge O-Level exams. In almost every paper they do ask about animal cell, plant cell, bacterial cells, their different parts, functions of their different parts, differences between their different parts and their levels of organization. How do these cells organize within an animal body, plant body or bacterial cells? And by the end of this chapter, they expect you to be able to name all the different parts and describe examples from animals and plant cells. And there are different questions that they want you to ask yourself. For example, why are different cells different in shapes? So for example, if we talk about a neuron is something like this. So there is a cell body and then there is a long process, and we will study about this. So a neuron is different from a skeletal muscle cell, which is something like this, and a skeletal muscle cell is different from a nephron, which is something like this. So all these different specialized cells, they have different structures. Why they have different structures? What are the jobs they do? What are their functions? How can we work out their magnification? Which means that if we look at these cells in the microscope, what do we mean by if we say we are looking at 60 times magnification? What does that exactly mean? So this chapter is about answering all these questions. And remember students, that in your Cambridge exam, concept is the key. If you don't understand any chapter by making concepts, you will not do well in your exams, because just ratification is not enough. Cambridge papers talk about logic, they talk about arguments. Therefore it is very important that you really understand the topic, you master the topic. Therefore I have decided to start each chapter line by line, page by page from the recommended resource, which is Hodders Biology. Okay, so let us now start. And once again, they talk about the focus points, that in this chapter what they will be focusing on is what are different structures and functions of plant, animal and bacterial cells. How do you identify cell structures in diagrams and images of animal, plant? Because in Cambridge exam they give you a lot of different images. So they will give you an image, for example, and they will have different parts labeled, such as part A, part B, and they will sometimes ask you what is part A, what is part B, simply their names. And sometimes they will ask about the functions, sometimes they will ask about more tricky questions. So you should be able to identify the image and diagrams. Okay, what are the differences between a plant and animal cell? Very important topic which we will study in this chapter. How are new cells produced? What is the mechanism of making new cells? Because you remember, in your body many of the cells are reproduced. For example, your red blood cells. So all of you have red blood cells in your body. The job of the red blood cell is to carry oxygen, and these red blood cells are alive in your body for about 120 days. After 120 days they die, and then the new red blood cells come into the blood. So we will talk about how they are produced. What are the specific functions of specialized cells? Some of the cells have very specialized functions. For example, some ciliated cells, some root hair cells, mesophyll cells in plants, palisade mesophyll cells, neurons in the nervous system, red blood cells, sperm and egg. These are all specialized cells, which means they are performing very highly valuable and highly specialized type of task. All this we are going to study in this chapter. What are the meaning of the term cell, tissue, organ system and organism? So these are the topics that we will study in this chapter. I will make multiple videos. Today's video is basically an introductory video in which I'm going to cover the initial couple of pages which are talking about cell structure and sections and staining and something about the microscope. Okay, so let us begin and start studying line by line what they have to say about cell structure. If a very thin slice of plant stem is cut and studied under a microscope, The stem appears to consist of thousands of tiny boxes or box-like structures. These structures are called cells. Now, first of all, let us try to understand what they are talking about. So, for example, if you go to a garden and in the garden you find grasses all around and you find somewhere a weak stem, you take that stem, and this, for example, is the stem of any grass from any garden. So that's a little stem, and somebody gives you a razor. Although do not practice this at home, unsupervised, do not practice it, okay? Always practice these experiments under supervision of your mentors. So if you have a cutting blade and you use the blade and cut it like this, so when you cut it like this, there will be two halves. So one will be this one, which is the lower portion obviously, and this is then the upper portion. And now the upper portion is like this and the lower portion is, so basically one stem you have cut into two different halves, half one and half two. Okay, now you take the blade and make a very thin slice, and now this thin slice, this upper bit, you take this and put it on a glass slide, and this is a very thin slice from the stem. And now you see this glass slide under a microscope. So when you look at this thin slice under a microscope, this is something which you visualize. You see there are small little boxes here. This box, this is a box. Each box is basically a cell. Each one of the box is basically a cell, and that is the job of the microscope, to magnify, to increase the size of what you are seeing. Basically, if you look at this small section or this small slide, you won't be able to see anything with your eye because these cells are very, very small. But when you set the microscope, in the microscope you have the luxury to change the magnification because there are lenses. We will, in the later part of this chapter, we will talk something about microscope. So a microscope has different lenses. For example, there is a lens which has a specific magnification of, say for example, times 10. Now this means that anything which you see from this lens will be magnified 10 times. So if there is a glass slide here and there is a section here and you are looking through this lens which has a magnification capacity of times 10, this thing will be 10 times magnified. So if the original size of this cell here is 1 millimeter, for example, what you will be looking at would be 1 multiplied by 10, so that is 10 millimeters. So see, you have increased the size 10 times, therefore you can easily see this. So that's the concept of the microscope, and it really depends upon what type of lens you are using. So for example, in this picture now there is another lens, and now I have the section which I am looking by this lens, not this lens, okay? And the capacity of this lens, for example, is 40 times 40. This little multiplication signs means multiplied by 40. So now if the same slide I am looking from here, which has the size of 1 millimeter, it will now be multiplied by 40, and you are looking the size of the cell 40 times enhanced. Is that clear? So the size, what you are seeing, the magnification basically depends upon which type of lens you are using in the microscope. So you should be able to understand this term magnification. Magnification basically means multiplying the size and to visualize things in a big fashion. So 40 times magnification means that the things are being multiplied 40 times. Whatever the original size is, multiplied by 40. 10 times magnification means multiplied by 10. Okay, so magnification, this is the terminology that you should understand. The other terminology that you should also understand is section. When in microscope world we use this word section, it basically means a thin slice of cells or tissue which is put on a glass slide to be seen by the microscope. So that is called a section. If I tell you this in a very common and layman type of terminology, so let's talk about this. All of you, prepare salads at your home. All of you prepare salads, I'm sure about it. So for example, there is a cucumber. So that, for example, is a cucumber. Excuse me for my bad drawings, but that will build your concept. So that's a cucumber. Okay, so if you cut the cucumber with a knife like this, and then you move your knife again like this, so you have this little slice of cucumber. So that is known as a section. Now try to peel off very thin section, as thin as possible. The thinner the section, the better the section is for microscopy. So you will be in a good position to see as many cells in a very, very thin section. Now you may be thinking that, oh, I don't get this concept, because you may be thinking that thicker the section means more the cells. Yes, that is true. But if there is a thick section like this, imagine there is multiple layer of cells, and with multiple layering of cells, the lens of the microscope will have trouble identifying each of the cell. So thin section means nice and crispy single layer of cells, and single layer of cells can be easily visualized by the microscope. So thin sections are good sections for microscopy. Okay, so you should have this concept of what is a section. So these three lines are very important. Let us read them one more time. If a very thin slice is equal to section, you call it either a slice or you can call it a section in biology. So if a very thin slice of plant stem is cut and is studied under a microscope, the stem appears to consist of thousands of tiny boxes. These thousands of tiny boxes are called cells. Okay, figure 1.1 is a thin slice taken from the tip of a plant shoot and photographed through the microscope. So what they have done here to generate this image is very important for you to understand. So how is this image generated? You should have the concept. As I said again, you go to a garden, there is a thin stem, you cut it and prepare a section. So now you have a very thin section of this stem. What you do is you put this thin section on a glass slide. So here is a glass slide and you put a very small thin section here. Now you want to see what is present in the section. You cannot see this with your naked eye. You cannot see that. So your eye does not work because these cells are very small. So you are very smart. You use a microscope with a lens, and say for example the magnification power of the lens is times 60. So whatever the size of the cells here, whatever it is, it will now be multiplied by 60 times and you look these big cells. So this actually results in this image. So now if you look in the microscope, now your eyes are not directly at the section, but you are looking here. So your eyes are here and you're looking through the microscope, so you will see something like this, a lot of cells. Okay, so that's the basic concept, that how this diagram is actually generated. Very good. So if you do not understand this concept, do not move ahead. Stop the video, see the video again. Okay, so you're looking at this image through the microscope. Now this is, they're telling you this is 60 times larger than life, which means that whatever the size on the glass slide is, the lens you are setting up for the microscope, because it is 60 times, so you're looking things 60 times larger. Now here is a little calculation here which is important for your exams. So if the cell now appears to be 2 millimeter, so say for example I pick up this particular cell and I tell you the size of this cell is 2 millimeter. Okay, now 2 millimeter is not the real size of the cell, because you are looking at the cell 60 times enlarged. So this is not actually the real size, 2 millimeter. If you have to tell the real size of this cell, what you need to do is divide this by 60, because this has been multiplied by 60. So I mean, let me go a little slow over here. So for example, here is a cell, teeny weeny very small cell. This is a very small cell. You are not able to see this, but you're seeing this in a microscope, and when you see in the microscope you see a big cell, and now the size of the big cell is 2 millimeter. Now my question to you is this the real size of the cell? The answer is no. This is not the real size of the cell, because the real size is very small. This is enlarged size. This is magnified. Now my next question, is this the magnified size of the cell? The answer is yes. My next question, what is the magnification to achieve this size of the cell? The magnification is 60 times, which means that you are seeing the cell how much time magnified? 60 times magnified. And after 60 times magnification, the size is 20 millimeter. So if you have to tell me the original size, it will be 2 millimeters divided by 60 millimeter, and the response would be the final real size of the cell actually is 2 divided by 60 is 0.03 millimeters. So a cell which was actually 0.03 millimeter in size, after the lens, which is enlarging it by 60 times, now the size you are seeing is 2 millimeters. Okay, now in examination they play a lot with this and they give you different scenarios. So for example, if they give you an image, and let's do it this way, if they give you an image that here is a micro photograph of a plant cell, and you are seeing, say for example, a lot of different cells like this, okay, and they tell you the size of this cell is 10 millimeter. Okay, they tell you this. So in exam usually they give you something and then they ask you something. So they are telling you that the size that you are seeing under the microscope is 10 millimeter. And now they are asking you what actually is the original size of the cell. Now in order to solve this, what you need to know is what is the magnification of the microscope. If they tell you that the magnification of the microscope is times 10, so all you have to do is divide the microscope size, which is 10 millimeter, by the magnification size, which is also times 10, and then you get the real size of the cell. So the real size of the cell is actually 1 millimeter, which means a 1 millimeter small cell after 10 times magnification in a microscope has now the size of 10 millimeter. Okay, so they can play around with these numbers. They will tell you some figures and they will ask you some figures. So be smart enough. If you understand the whole procedure, that what's happening, how is the microscope magnifying, then you are able to solve this. Okay, so I hope this paragraph is very clear to you. Students, in my videos at this YouTube channel, I will focus on each and every line of the book, and I want you to focus on each and every line of the book, because if you understand with concepts, you will easily crack the exam. Okay, now figure 1.1, this is the figure, this is a longitudinal section through the tip of the plant. We have discussed that, which is times 60 times magnified. The slice is only 1 cell thick, so light can pass through it and allow the cells to be seen. You remember I was telling you here that if you have a thick section, if there is a thick section, it's not a good section, because there are so many cells and light cannot pass through. But if it's a thin section, it's a good section. Okay, all good up till now? Now we have to understand two terminologies. There we have used a terminology called longitudinal section, and you should master what do we mean by longitudinal section. Okay, so here for example, thin slices like this are called sections. And we now know this, that slices are equal to sections. So if you take a blade and cut something like this, so this piece, this section generated is known as a section. Okay, now there are two ways to cut something, and they are both shown is in this particular diagram. So there is this cylinder like body which you are cutting in this way, so the section that you will obtain will be something like this. So this is what you will obtain. Okay, so this is known as a transverse section. What do we call it? A transverse section, because you are cutting it horizontally. But if you cut something in a longitudinal fashion like this, so now this is no more in the standing position, this is now in the lying position, and you are cutting it in a longitudinal plane. This is known as a longitudinal section, and this you can see and practice in your everyday life. For example, again, example of a cucumber. So suppose this is a cucumber. Some people cut cucumber in longitudinal fashion like this. So the slices that they generate are actually long slices of cucumber like this. So this is known as a longitudinal section. But some people are fond of cutting the cucumber like this, like this. These are the slices, so they get round slices like this. Okay, so this is known as a transverse section, and this can be applied to anything. Think about carrots. I mean, if you have a carrot there and you cut the carrot in this way, this is a longitudinal section. But rather, if you don't cut it in a longitudinal way, you cut it like this, then this is a transverse section. Okay, so a transverse section is a horizontal section, and a longitudinal section is along the long axis. So these two terminologies you should be very clear about. So if you cut along the length of the structure, you are taking a longitudinal section. But if you are cutting it across the structure in a horizontal fashion, it's a transverse section. So these two terms are important to know for this paragraph. Okay, so now let's move on. You can cut sections through plant structures quite easily. just by using a razor blade. Now that's important thing. So if you are dealing with plant structures such as plant stem or cucumber, which is a plant, or potato, which is a plant, or tomato, all these are very easy to cut because you just take up the knife and you start cutting them, very easy. But cutting sections of animal structures is more difficult. Why? Because animal structures are usually very soft and flexible. For example, liver, for example, muscles, for example, kidneys. If you want to cut them, it's difficult to cut them because they are very soft. They move here and there. So in order to prevent such movement, what we do is we use a wax. So here is the concept. Focus on this very, very carefully, okay? Now, what we will do is we will talk about a piece of kidney, for example. So here we have kidney, and kidney is like this, bean shaped. Now we want to cut kidney. If we want to cut kidney in a longitudinal plane, that will be a longitudinal section. If you want to cut kidney on a horizontal plane, that will be a transverse section. But regardless of whatever section we want to cut, cutting kidney is difficult because it is a very soft structure and it keeps on moving. So what we do this. We use a container and put a lot of wax in the container, and that wax is molten form. It is actually, we melt it first. This is molten wax, and this molten wax fills it up till here. Okay, so this is now all filled with wax. Now wax settles down when it is no more in the liquid form. It solidifies. What we do is we take this out, and the container is gone, and now there is a kidney, and this kidney is all surrounded by a solid wax, and this solid wax is easy to cut. Now you take the knife and cut it all the way through. So basically you are cutting through the kidney, but now the kidney has been stabilized by using a wax. Okay, so and later on when we do different procedures, we want to stain it, we want to color it and see into the microscope, the wax melts again and goes away. So you actually introduce the wax just to make the cutting process easy, and now the wax is gone and you are left only with the section of the kidney. So basically what I'm telling you is for animal cells you usually use wax so that the organ becomes a little solid and thin sections are easy. The wax is dissolved away after making the section. It goes away. Okay. Now, when sections of animal cells are examined under the microscope, they too are seen to be made up of cells. But the cells of animal tissues are much smaller than plant cells, and they need to be magnified more, sometimes as much as 700 times, you see. So for example, if you have to see the cells of the kidney, you have to use a powerful lens, a lens which has the magnification capacity of 700 times. Okay, the sections are often also treated with dyes. So sometimes some of the cells are basically transparent, or they do not have any colored structure. So when you see under the microscope, you actually don't see anything. Therefore we add some color to it. So since we have added colors, this is called dye using, or another word for this is staining. So when we use this word staining of cells, that basically means we are adding some color to the cell so that we can see the cells more clearly. Okay, so we have introduced a lot of terminologies for you in this chapter. You should know what is a horizontal section. You should know what is a transverse section. You should know what is magnification. You should know what is the power of the lens. You should know how to work out the magnification. You should also know what is staining. All clear up till now? Okay, let's move on. Making section is not the only way to study cells. Now that is a very, very important conceptual type of paragraph. Thin strips of plant tissue, only one cell thick, can be pulled off the stem cells leaves directly. Plant or animal tissues can be squashed or smeared on a microscope slide, or treated with chemicals to separate the cells before studying them. Now, let me explain this paragraph to you. Up till now, we have been talking about sections. I told you if there is a plant tissue that we are cutting, we simply take up the blade and start cutting it. If there is animal tissue, we have to use wax and maybe sometimes stain them. But some of the tissues are different. For example, if we talk about spleen. Now, spleen is a very fragile type of tissue. So if you simply have to see the cells of spleen, what you do is you use one glass slide and there is another glass slide, and between the glass slide you crush the spleen. So when you crush the spleen without any wax or without anything, cells from the spleen, they are released and they spread on the slide, and you see this under the microscope with the use of a lens. So here what you are doing is you are not using any wax, so there is no wax needed. No wax, no. There is no razor or blade needed to cut spleen. No. You simply crush it. You crush it and you see the cells. So this paragraph is actually telling you that there are some other ways to see the cells as well. For example, you simply squash them or you simply crush them. Okay, so that's an important thing for you to understand. Now, there is no such thing as a typical plant or an animal cell. Many students use this terminology. What is a typical plant cell? What is a typical? So there is nothing like a typical plant or animal cell, because each cell basically vary in their size and shape, and this depend on their function. For example, the cells of kidney are different in shape and size from the cells of nervous system. The cells of heart are different. The cells of stomach are different. So each cell has a different function to perform, and therefore they have different shape, size, and distribution of different structures within them. Therefore, do not use this word a typical plant cell or typical animal cell. Okay, every cell is different. However, in order to make things easier, we usually use this drawing. This drawing is used, which is usually a circular drawing. When there is a membrane out there, this is called the cell membrane. Then there is a rounded body inside which contains the genetic material, the chromosomes. This is known as the nucleus. And anything between the cell membrane and the nucleus is known as the cytoplasm. So these three terminologies are something that are extremely important. We will study about them: cell membrane, cytoplasm, and nucleus. But remember, this representation, the round representation of a cell, is not applicable to every cell of your body. For example, I told you that a neuron has a cell body and then a long axon. A nephron is different. A cardiac cell is different. So structure and size will be different of different tissues depending upon their job and functions. Okay, but in general, they have almost all of them have a cell membrane, a cytoplasm, and a nucleus. If we are talking about animal cells, the plant cells have some additional layers. Okay, and things are different in bacterial cell. This is an example from the animal liver cell, right? Okay, I think I will stop the video here. In the next video, I will start off by discussing cytoplasm, cell membrane, and nucleus. Up till now, this is I think a lot for this video. Watch the video again and again, share with your colleagues and friends, and subscribe the channel. Welcome to my channel and stay tuned for more videos. Take care of yourself.